that's it. And with this, I will start the presentation. So today we will be talking and learning about the functional annotation and analysis strategies we have available in OmicsBox, and which are done with the Blast2GO methodology. So before starting, please let me present a little bit the BioBAM and our company and our software solution. So I will introduce BioBAM and OmicsBox, and then our goal is to learn about functional annotation concepts and to show the functional analysis options within OmicsBox in detail. And then we will go into the live demo session where I will annotate and analyze an RNA-Seq bulk dataset. So about BioBAM, so we are a bioinformatics solution provider with a team of experts in terms of bioinformatics, but as well as computer science. And we have this mission to close the technology gap between the computational bioinformatics research and the molecular biology research, with this main motivation to make bioinformatics a little bit more easy for all of those which do not have a bioinformatics background, but still need to do bioinformatics tasks to get their research, to progress their research. So our solutions, in particular OmicsBox, is used by several hundred international clients all around the world. So there's a lot in the US, but in Europe, Asia, Australia, and India as well, of course. So OmicsBox, we like to name it like an end-to-end bioinformatics platform, mainly for next-generation sequencing data. And it's actually a desktop application which works on Linux, Mac, and Windows. And it can be installed in your desktop, and it's then used for bioinformatics applications, mainly for basic research, biomedicine, agrofood ecology, or biotechnology. It's pretty popular among researchers who are working with non-model organisms because it's very flexible in this sense, and you can always, like, without prior knowledge, get into OmicsBox, provide the information you have, and then try to find functional insights from there. So this is OmicsBox. So OmicsBox is split or is divided into bioinformatics applications, which we call modules. And there are actually right now 5 of them. So it's genome analysis, genetic variation, transcriptomics, metagenomics, and the functional analysis module, which we will explore and see today more in detail. Before we start right with this, I want to briefly go over a few slides of these other modules just for you to know what you can expect there. So for the transcriptomics part, we are able to analyze bulk RNA-Seq, single cell and long-read transcriptomics data. And the pipelines or the options you have look a little bit like we tried to show in this scheme. So when you work with long reads, NanoProBac Bio, you can do alignments, you can do transcript identifications, then do reconstructions, quantifications, and then the expression analysis, and then with SCAN-T3, the transcriptome curation. Similar for bulk, for single cell data, you have the quantification of UMI-based or 3' or full-length data, doing clustering, cell type identification, trajectory invariance, and then the interactive visualization of these different types of projections of single cell data, and then again going into difference expression and the functional analysis, always trying to go end to end, like from the raw data to the functional interpretation of those type of data sets. Same for the genetic variation module, where you go and use your raw data, and then you do variant calling, filtering, annotation. You can go into gene-wide association studies with the functional interpretation again here, or additional tools like the population structure analysis with admixture here at the bottom. Metagenomics, similar, you start with raw reads, you do, like in most of the steps, you do a quality control and assessment with FastQC and Trigonometric, and then you have the option for the taxonomic classification or the assembly and gene prediction of metagenomics data, and then you compare different samples or That means you compare it by the composition of taxa, differential abundance taxa testing, or you have the comparative analysis of functions, which is kind of the functional interpretation or comparison of different samples for the metagenomics data you might want to have. And then the functional annotation and analysis module. There are many, many things there, and I'll try to cover quite some today. So here you start with a set of sequences, which could be nucleotide and amino acids, normally in FASTA format. You can also import them. And then you have a whole set of different algorithms you can use to assign functional labels to your sequences. And then it gets very colorful. There are a lot of different visualizations. And once functional annotations are merged and annotations are done, you can go into the functional analysis. And there you have enrichment, pathway analysis, quantitative analysis, or you can go and do certain data manipulations, which are often needed, like a batch renaming or ID mapping or selecting certain parts of your data filtering, and so on and so forth. Okay, so when talking about the functional annotation in OmicsBox, we need a lot of external resources there. And by resources, I mean databases and algorithms. And they are all readily available within OmicsBox. So obviously, the most important one is gene ontology. And then there are important algorithms like BLAST, Interpro, Diamond, AFAM, and so on and so forth, as well as functional analysis methods like GZA, database systems, and so on. So this is all available within this from within this graphical user interface, which I'll explain in a second. But maybe first, something about the functional annotation process itself. It's pretty basic, but maybe it's good to remind this. So normally we start with uncharacterized sequences. That means genes and genes or proteins or transcripts. And those for those sequences, we perform certain algorithms, which means we align them. We look for protein domains or families, or we can go and search for autologous groups with algorithms like Mapper. And from there, we link, or with those, we link functional information to our original sequences, and that's the gene ontology. But linking is not enough. We need to transfer this information and validate it and curate it and make and remove redundancy and make it consistent and apply to the true path rule of the gene ontology to have only the most specific functions to our sequences, and all that is done with the Blast2GO methodology by doing this transfer of functions back to our uncharacterized sequences. Okay. So, and this is not a one-click, one push-button application within OmicsBox to run just Blast2GO. We see it more as an interactive toolbox for adaptable functional annotation strategies. And there we have different steps we perform, one after another, or at the same time, or in parallel. And then in each individual step, we have a lot of different charting options and different result visualizations where we can review on a dataset level or on a sequence level, like how things went. Okay. So here we can see Blast and Diamond, things get orange. With GO terms mapped, it gets green. When functional annotation is present, it gets blue. Interpro and Eggnog, additional annotations, additional sources of functional information, which can then be merged onto our project. And then the graphical user interface, which allows us to navigate all of it. So before getting into the demo, I actually want to spend just two little slides on the graphical user interface so you can already find yourself a little better while I'm going through it. Though this is OmicsBox in action. And here we always have a top part where we have our data tables with our side panel, very important, where we find downstream analysis options. And then in the bottom part, we have the different visualizations. So here we have an interproscan result, gene ontology terms with their descriptions, a gene ontology graph, and a BLAST result with their alignments and their detailed information. So these are the things we can get throughout the annotation process. And there are two things we use most. One is the side panel, where we can go step by step from BLAST to Interpro to mapping to annotation, for example. And then don't forget about the tools and selection functions. And then the context menu. So for each individual sequence, I can right-click, and then I will get the context menu, and then I can show the sequence, show the BLAST results, show Interpro scan results, and so on. Okay, so here are additional things like create ID lists, create charts, copy cell data, and basic stuff you need for data manipulations. Okay, so an example workflow in OmicsBox, and I think which best represents the Blast2GO annotation strategy, might be something like this. These workflows are available within OmicsBox, so you can and they help to understand how things can be configured. Today we're gonna analyze each step individually, but just for you to know. So we start off with a FASTA file, then we run Diamond, Blast, Interpro, and Eggnog in parallel. And then we do geo mapping annotation, and then we merge data back together over here and over here. Then we validate our annotations, and finally we have our annotation project, and then we can export those annotations into certain different custom formats or visualize data. So this is like the default or the standard strategy, which is quite complex. Please note that there's a lot of different annotation strategies you can perform within OmicsBox, and that's just one possible option. Okay, so a lot of people do like to go just for Interpro. They go just for protein domains and families, and then they do transfer gene ontology function from there. Others like to take into account evolutionary concepts, orthology groups, phylogenies. So they will go for Eggnog, and they will transfer functions from the Eggnog. Maybe the most conservative approach would be to just go for the top BLAST hit. So that means take my uncharacterized sequences, do a BLAST search, and then look for the first hit which has a gene ontology term, and then transfers the functions from there. So, and in between, there's a lot of other options. So you can go and do an iterative approach. So go for one type of analysis first, annotate the first part of my data set, and then add additional information to the sequences which are left over. And then I can go from permissive from a strict to a very permissive annotation. That means my coverage. So if I have more or less sequences of my dataset annotated, change. But I can also adjust the annotation to go for more general to more specific ones. So that means I will stay more in the top part of the gene ontology, general concepts, or I try to go as specific as possible, always trying to annotate to the most specific geo terms. So there's a lot of different things, and I will try in the demo. I will try to explain some of the parameters which adjust those, or which realizes which helps to go for those different strategies. Okay. So once our data set is functionally annotated, the next step would be the functional analysis, obviously. And there, the most common thing is to do an enrichment analysis. And there we have two different options. So we can compare two different lists against each other. And then normally people go for the Fisher's exact test, which uses a contingency table, counts how many sequences are annotated to a certain function, and then compares with whether if they are more abundant in one group of our list of IDs compared to the reference, which is sometimes or most often the rest of the genome. Alternatively, and when we are having a gene ranking, that means, for example, if we have a difference expression data analysis and we have a ranked list, which could be a log-fold change or a statistical value like an FDR, we can go and do a ranking. Though that means here we don't have to have a sharp cutoff saying this is overexpressed, this is underexpressed, but we can just have a ranked list, and then we do a gene Z enrichment analysis. And there we are kind of have the annotations linked to our sorted list of genes, and then we find if there are certain functions which are more frequent at one end of our list compared to the other end of our list. And this is all things you can do in OmicsBox with just one click of a button. And once those analyses are done, there's a lot of different visualizations for functional information, for gene ontology in particular as well. So here we can find word clouds, which are pretty fancy. We have the old-fashioned kind of bar chart where we can see functions which turn up. We can look up things on the gene ontology graph, and there we often see that there are many enriched functions who lie on the same path, and there's a specific function to remove those. So we'll stick in our enriched function list to only the most specific ones, which is sometimes helpful if you get very long lists of functions. And then we have the bubble plot, which kind of got popular more recently, where you can see like the size of like the amount of sequences associated to a certain function represented by the bubble size, and the statistical significance by the color. So things come in handy as well. And then last but not least, in OmicsBox, we have this nice combined pathway analysis, which allows you to go in with your sequences, just sequence data, and or particularly interesting the or, because you can just go in with sequences without annotations, or you have an annotated set of sequences, and you can combine it with your expression data. So here, why is it called combined pathway analysis? It's called combined pathway analysis because we have different database systems like Reactome, KEGG, Gramini, and then we combine those in one analysis together with the difference expression and the annotations. We perform an enrichment analysis, and then we get this nice visualizations where we have enriched pathways with expression profiles, and we can review our data here. So this is how it looks in OmicsBox. You get this table with different pathways which are statistically significant, and then on the map you can see expression profiles and your experimental design. And then you can see that certain enzymes linked to certain transcripts, for example, with a statistical significance, are differently expressed in case versus control, or in control versus case. Okay, so these are things which we will explore in a second in live with OmicsBox. And this brings me to our use case for today and for the OmicsBox demo. So we will be using basically only two modules today, which is the transcriptomics module for our raw data analysis, and then the functional analysis, where we will do the annotation and the functional interpretation. Okay, so the dataset we will be using today is a bulk RNA-Seq dataset from a worm parasite, which is called Ascaridia galli. It's actually a nematode roundworm which affects chickens and makes them really sick. So we have two types of chickens. One were treated with flubendazole to kill the roundworm, and the other ones not. And so the roundworm parasite had been sequenced with 12 FASTQ files, which is actually paired end data. So it's actually 6 samples. And this is the data we will be using of Ascaridia, and we will not have anything else. No reference genome, no reference annotation, just the raw 12 FASTQ files from this publication. So it's actually a publication from +1. And this is the experimental design. I picked something fairly easy for today. So we will just have 3 samples with control and 3 samples with treatment. It's about 2 GB each of those files. It's paired end, and it's about 25 million reads per sample. Here you have the link to look into this paper as well yourself if interested. You can also find it in our help center under example data sets, where you have like the whole analysis pipeline and the intermediate data files and results as well. Okay, cool. So with this said, I'll get out of the presentation mode and I'll open up OmicsBox. and we will start presenting things, showing stuff. Here we are already. This is OmicsBox opened up, empty, and we can see here our progress. There are already a few things which I just remove. And then we have our local files and our application messages. Okay. And then I will just go and show you our raw data. So we have here our 12 files of FASTQ files, which we'll be using throughout the analysis. So before going into the functional annotation, I will show you where those sequences were coming from. So it's actually those 12 files. And we were taking those files, and obviously we're going into the general tools, FASTQ tools, and we were checking the quality of those reads with FASTQC. And we got this kind of 12 rows in our table where we get a review tag for all of them. We could see again some details of the data set. And when I click on it and show report, I can see an individual report of each of those, and I can see that some of those tests failed. So my quality, for example, passed, but I had some adapters which should be removed. So what I did next was I went into FASTQ tools Trimomatic, and then I provided here again my my files. I selected parent, I selected all my samples, and I clicked next and said remove adapters, add trimming, quality trimming I did not do here, but I did filter by quality and filter by length, and then I just hit run. And this gave me my clean reads over here, which I then used for my transcript assembly. So as you can see here, I always get those little reports when doing something in OmicsBox, and which can be exported as PDF as well. And here I can see my results overview, input reads, surviving reads, things which had been filtered out, some analysis parameters, and the corresponding references here. So with those clean and filtered reads, I then went into the transcriptome characterization, and I followed basically this path. So Trimomatic, FastQC, and then I did the de novo assembly. I did some clustering with CD hit. I checked for the completeness assessment of my assembly with BUSCO, and then I did a prediction of coding regions. Okay, so this can all be found in the RNA-Seq assembly over here, where I did completeness assessment with BUSCO, predict coding regions, sequence clustering with CD hit, and so on. And this analysis brought me actually to this FASTA file, which I will open up, which turned out to be 62,000 CDS sequences, well curated, I would say, by following those analysis steps within OmicsBox. So I'll close those things and also close the reports over here. And I'll click and open those files. So I will say load sequences, and I will open them. And then I'll get this progress over here. And then I'll have my raw sequences here. So now you can see how this works. We have our 62, you have always some table entries over here, it's 62,000. And then I can right click on my context menu, I can go for show sequences, and I can see that it's a nucleotide sequences. And then my first thing here would be to start with a blast search. So when being over 4,000 sequences, I always do recommend a Diamond Blast for performance performance reasons. And here we choose Blast2X because it's it's nucleotides, more sensitive is fine. And we are going for the NR database. But since it's a very huge data set and we don't want to go blast our sequence against all the bacteria of all the world, we will just go and select a taxonomy filter of nematoda, an expectation value of 3, a number of blast hits 20, and then we will hit on run. And this starts our blast search. And this means sequences are getting sent away and blast results getting recovered. And then we will get our results into this table. So in parallel and at the same time, we will also start an interproscan search, which allows us to search for protein families and domains. So we go cloud interproscan next, and then we choose all the different algorithms we are interested in. So there's superfamily, there's pfarm, there's funfam, and so on and so forth. And then we hit next. We decide on whether if we want to have some particular export formats or if we will just leave this blank, and we hit run. And I did this already. And then we will see how this looks like in a second within our project. So now Blast and Interpro are already being executed in the cloud, and results will get back into this table within a second. Additionally, since I want to follow this full pipeline with all 3 type of algorithms we have available at the moment here, I will also go and run the functional annotation with eggnog. The eggnog mapper has to be executed from the main menu over here because it's kind of apart from this project. And you provide here your, again, our 62,000 CDS sequences. And then we just hit run, and this executes eggnog. There are no further parameters for this step. And then with this, we have our functional annotation already on the way. But maybe before going more into detail of this, I wanted to show you in terms of the difference expression analysis of this data set, how things went. So in the transcriptomics module, we performed our assembly and we got our transcripts. Then obviously we needed to do our quantification. So quantification can be done in OmicsBox by gene level quantification or by transcript level quantification. So since we do not have any reference genome and any reference and any structural genome annotations, which means TGF, GFF files, we went for the transcript level quantification with an algorithm called RNA-Seq. And here we just have to provide our raw FASTQ files and then kind of our transcriptome. So the transcripts we just assembled with Trinity. So we provide FASTQ and FASTA. This looks like this. Again, we choose paired end. We select our quality assessed and trimmed sequences over here. We select our upstream and downstream patterns. And then we go for the and then we provide our transcripts down here. And then we just hit next. We can check, we can select strand specificity and some other parameters. And then we will just hit run and we will get our quantification. So I will open up the result of this process, which gives us basically a count table. So here we can see our transcripts again. So there are the same IDs as we have in our annotation project. And here we can see we have our sample names in the top column, and then we can sort and filter those columns. And we can see here already our reads, which are raw reads quantified here. Then in the side panel option, we have a lot of different charts to explore this part of the process. So this table, so we have the library size per sample, or the distribution of counts, distribution of library sizes, and even a PCA plot to see how those things cluster together. So I can check and see that the distribution of our samples are more or less fairly equal. And with that, let me see this plot. Yeah, I get also this report, transcript level quantification, where I can see again my samples and how the analysis went here, how many sequences could be significant positively aligned, mismatches, and so on and so forth. So this quantification now I will use in the from the side panel action here to perform a difference expression analysis. So I will click on differential expression analysis, and I can go for a pairwise without replicates, and the time course. Since I have a very simple experimental design for today, I do not have time points here. I'll just go for pairwise, and I will click next. And then I can choose my counts per million filter. So for example, if I have 12 million counts, and if I put this to 1, I have to have at least 12 reads linked to a certain sample. And then I'll put this to 1. A good value for the samples reaching the KPM filter is like the size of one of the experimental groups. So since I have 3 samples with control and 3 samples with the drug, I will set this value here to 3. And then I hit next, and then I'll provide my difference expression design. So I'll just open that here. So I have just my sample names over here, and then my treatment and my control tags or metadata on the other side. And then I'll go for a simple design, and I say I have my treatment condition, because in this experimental design I have just the treatment, and I'll say select as contrast flubenazole and as reference condition my control. A statistical test would be an exact test, for example, with this robust option of HR, and then I just click the Run button, and I will get my pairwise difference expression results back here. So this table looks slightly different. First of all, I have my text. So if I select up or down regulated sequences and sort by the FDR, I can see here my up and down regulated transcripts. And from here, I could directly go into some functional analysis options, like, for example, the Fisher's exact test or the gene set enrichment analysis. But maybe before doing that, we can check a few other things, like, for example, the volcano plot. In this plot, for example, I can see that when setting my up and down tags to logfold change of 5 and to an FDR cutoff at this level, I can nicely pick the most differentially expressed genes marked for down and up regulation in green and in red. So just for you to show, the up and down text can be adjusted here, where I can say like p-value FDR, the value logfold change 5 and 4. And if I go and adjust those values, then my text will be changed. And when I recreate this plot, then I will have a different visualization here. And I can also have a quick look at my summary report, where I see the pairwise difference expression results, the number of total features, upregulated, downregulated ones, my experimental design, and the analysis parameters I used for this analysis. So with this, we are then now already ready to go and continue working on our functional annotation, and then do the functional analysis. Okay, so the functional annotation, I said we were using this raw FASTA file of our CDS sequences, and we performed BLAST, interpro, and eggnog. And now if I open up, for example, this file, I can see how those white sequences turned into, I'll close some things. So this will take a second. So this is 280 megabytes of blasted and interpro scanned sequence data. So it takes a second to open those. So now here I can see my sequences turned red or orange depending the on the success of the analysis, let's say. So if I pick, for example, this one here, I can see I have interpro scan IDs over here. And if I right click, I can see that both results are present. And then I can check my blast results. Then I can check my alignments and check like the details for each individual sequence. And then this is used to perform the gene ontology mapping and the functional annotation. So the interpro results look something like this. And again, here I can see different types of algorithms, families, homolog superfamily domains with different gene ontologies, which are consistent here from different source databases performed by interproscan. So now the next step would be to perform the gene ontology mapping. So I just pick one of those, like the latest version of the gene ontology database, and I hit run. And then I will get the gene ontology terms linked to those sequences. While the mapping is being executed, we can take a look at our ECNOC results, which we also started a few minutes before, and which should be now, which we can show now here. So ECNOC is kind of a different type of project because it has different tags but we have our query IDs, which match here again with all of our other projects. That's very important. Because then we can link things throughout the analysis. And here I have, again, e-values and bit scores, and I can see from which taxonomies those functions have been transferred, and functions can be shown over here. So now a very important step in OmicsBox, once we have our sequences functionally annotated, which means mapping has been performed and the functional annotation has been performed. Let me see. Then we will go and hit the gene ontology annotation step. And here we have a few parameters which are important. We can do first filter our source blast results again by providing a different cutoff for our hits, for our e-value, or for our highest scoring pair hit coverage. That means the percentage of the sequences being aligned from the query and from the hit. And then we could, for example, go for a top blast hit strategy, which means putting this to 1 and only taking the top hit with a GO term. Or we can just leave it as default, go for 20 hits, and then use the annotation cutoff here, which kind of takes into account the sequence similarity, and the GO weight takes into account where at the gene ontology hierarchy we wanna annotate. So if I have a lot of source hit annotations at the bottom, those sum up going along the gene ontology, and then I will pick one of those where I have more evidence. Next in the parameter settings are the evidence code weights of gene ontology terms, and here I could, for example, filter out automatically assigned evidence codes by putting a value of 0 here. But this normally leaves you with very few functional annotations, because most of the functional annotations in public databases are actually coming from those electronically annotated ones, okay? And then I hit run, and then I'll show you how this looks like. By opening up an annotated dataset now. Obviously, what I didn't show is for each of the individual options, you can go and review intermediate results. So there's a lot of different charts for BLAST, e-value distribution, top-hit species distribution, and so on and so forth. It's a similar thing for the InterPro-Scan, a lot of different types of charts, as well as the gene ontology mapping. So you can see here geomapping distribution, enzyme code distribution for sequences, and so on and so forth. And then the most interesting ones, I guess, is the geo annotation statistics, where I can see how many geos do I have per sequence. For example, or at a certain gene ontology level, how is my distribution among the different gene ontology categories, and so on. So now in this table, I can see my final annotation project. So I have those sequences which do have interpro results, BLAST results, which had been gene ontology mapped and which got successfully annotated. And when I right click here, I can see that I can also review now the mapping results and also the gene ontology descriptions. For those who really want to know how the algorithm picked those annotations in the end, you can also look at the geo mapping graph with annotation scores, and then you can have a very good understanding of the actual annotations and how they had been performed. So now maybe you might wonder, if I would have done annotation just by Interpro, and if I would have done annotations just by BLAST or just by Magnoc, I prepared this kind of little Venn diagram on this data set. And I did that basically by just going into, for example, this project and going into the type, into the like filtering for the ones which are annotated, and then extracting with a right click, creating an ID list of this column of annotated sequences. And then we can see here, if I make it to proportional, we can see here that InterPro-Scan annotated 33,000 sequences, BLAST annotated 17,000, and Eggnog 15,000. And all combined, I have now what I have in this table, where I finally went into the functional analysis, functional annotation, and did the merge Eggnog annotations. And I went into Interpro and I did merge GOs from Interpro, and everything got together in this final annotation project. Okay, so this is the final annotation project, which can now be used for the functional analysis of our results. And here I have several options with OmicsBox. So the first option would be from the annotation project, going into the enrichment analysis, and then performing a Fisher's exact test or a gene set enrichment analysis. So when I do that from within the project here, I have to select a test set, which is kind of the up or down regulated gene IDs I got from my differential pairwise difference expression result over here. And then the reference set can be defined, or otherwise it would be the whole list of my annotated genome over here. Then I can go for two-tailed to find over and underrepresented functions, and I can select which functional labels I want to check for and which gene ontology branches, for example, I want to do test for. So this is the Fisher's exact test, where I have to compare those kind of two different lists. For the gene set enrichment analysis, it's slightly different, and I have to provide a ranked list. So how do you get this ranked list? You get it also from your pairwise difference expression analysis, or from some kind of other analysis you had been doing. And here again, you can choose molecular functions or biological processes. You can go and filter for your FDR, and so on and so forth, and you'll get your results, which we'll review in a second. But before going into that, I will show you a maybe more easy, a more straightforward way to perform the functional analysis in OmicsBox, and that is when you have already generated your differentially expressed data here, you can directly go from the side panel and go a Fisher's exact test from here. So, and this allows you to just automatically run against up regulated genes, for example, your test. And then at the reference annotation, you would have to select this blue project we just generated, okay? And this is the annotation. This is just the box file you store when storing this project here. And again, then the other options stay the same. You have two-tailed, you define the type of annotations you want to test for, and you hit run. Similar with the gene set enrichment. Here again, the ranking will be formed automatically by combining the log-fold change with the p-value. And then the reference annotation has been provided manually. Okay. Then the analysis options stay the same for the gene set enrichment analysis. So we hit run, and then I'll show you how those kind of results look like. So now we performed the assembly. We performed the difference expression. We did the functional annotation of our data set. And now we will go into the functional interpretation of the functional analysis. So first we did a GSEA, a gene set enrichment analysis. Oops, I dragged this down here. And now in this table, we will not see sequences or transcripts or genes or proteins here anymore, but we will see geofunctions with their name, okay? And then top and bottom means overrepresented in the overexpressed part of my list, and bottom means overrepresented in the bottom part of my list. And for each of those functions, if I right-click, I can show the details. And then here, for example, I have ubiquitin binding. And then I can see here the distribution of my expression values of my ranked gene list. And then I can see that there are more upregulated genes which are associated. Associated means like this little black bar, which are associated to this gene ontology term in this area, and less in the bottom part of my list, okay? And now if I would go into a bottom one, I should see something the opposite. And here I can see that, for example, this extracellular ligand gated, it's a molecular function, is more frequent in the down-regulated part of my ranked list, and that's where I get this enrichment plot the other way around with statistical significance. So maybe interesting options here, apart from the details of each of the specific functions, is the bar chart, the bubble plot, the word cloud. I'll just open some of those here. So here we can see the word cloud. Maybe I can click. The bubble plot is also coming up. Maybe I can open up this one here, which is already kind of ready. Right, and then I can like filter out, I can modify the bubble size, the font size, how many elements I want to show, and so on and so forth. Okay, so this is like obviously those kind of labels can be changed, and then there's a lot of different options to review the results here. Sorry. Yeah, and this gives you, here we have to look for the normalized enrichment score for significance, and this gives us the list of gene ontology functions. Most dominantly, what stands out is kind of this ubiquitin binding for our functional analysis. Similar, the Fisher's exact test. I will not really go too much over those results, just for you to see that here we have also overrepresented functions. with our p-value over here, and then we have similar charting options: bar chart, bubble plot, enrichment, go chart. Maybe just a quick look on this gene ontology visualization, which is kind of neat. So here you can see enriched functions on the gene ontology graph, and then you can see that there are certain functions who are who you can find along the path, which you could also filter out to have a shorter list of enriched functions for your Fisher's exact test. Okay, those are the classic kind of enrichment methods. I really want to show you also the combined pathway analysis in OmicsBox while getting to the end of our presentation here. So here we provide sequences with annotations or without, and then we combine it with different expressions. So everything which we have seen here today, we will use. So we will use our pairwise result, we will use our annotation table, and then we will put all this into a pathway analysis. It's a combined pathway analysis. So I'll just pick here my functional annotation. So I have blast, interpro, annotate, merge with eggnog. I'll open this, and then I'll check and do my difference expression result. I have my pairwise enrichment here, and then I will combine this with that, and I go for a pathway enrichment analysis, and then I have to pick the databases I want to run this against. So I have the Reactome, I have Gramini, and I have KEGG. And for each of those different pathways, you have different linking options. So if I would not have done the functional annotation beforehand, I could directly, with just plain sequences, do the linking of my pathways via eggnog within this dialog here. Since I have that, I can deselect that now, and I'll just use enzyme codes of my functional annotation project. Okay, and then I hit run, and this takes, depending on the size of the project, takes like 20 to 30 minutes, and then I'll get this kind of pathway analysis project with a given in table format. And here I can see which database, KEGG Reactome, which pathways, the corresponding IDs, from which sequences, from which species they are, and then the amount of sequences which are linked to a certain pathway. So I already can see thiamine metabolism, purine metabolism, they're linked to many, many sequences, but I'm actually interested in differentially expressed sequences. So I have the galactose metabolism. There's 60 sequences attached, 4 are differentially expressed. So and now I go show pathway diagram. And I will make this a little bigger for you. And now I can visualize the galactose metabolism pathway with all the linked sequences marked in colors. And with the side panel option here, I can just show the expression values on the map. And then I can go even further and say, oh, show only the results with differentially expressed data. And then I can go and see that those type of enzymatic activities, sucrose to fructose, for example, here this enzyme had been upregulated under control and downregulated under drug condition. And I can see like to which sequences or transcripts this enzyme is linked in. And here it has been, for example, it's the exact opposite way, downregulation in control and upregulation with flubendazole exposure. And here I can see on the side panel all the different results once more, and I can switch them on and off and do my explorations by searching for different concepts. I can actually also search here at the top part of my pathway project for pathways of interest. For example, when looking at flubendazole and the effect on parasites, we might know that there's the microtubulin formations which are affected by this drug, and then I could look for specific pathways which affect those kind of structures and how this drug might have had a very detailed effect, or which genes or transcripts had been involved in affecting the formation of those structures in the cell, for example. Okay, and with this last part. I think I'm at the end of my demonstration, and I will quickly get out of OmicsBox and back into my presentation. Okay, so I think the most important take-home message today, which I wanted for you to know, is that we managed, like within this setup, to get really from just a couple of FASTQ files, from raw reads, to a defined transcriptome, to the functional annotation of those transcripts, and then into the functional analysis and interpretation of my experimental design and of the experimental data. So I could really get from end to end, from raw reads to functional insights for a non-model organism where they had no reference genome, no reference annotations within this setting. Okay, so combining like the transcriptomics features and the functional annotation and analysis features. You could see that there's a lot of different tools and algorithms and databases had been involved here, and they kind of are all nicely integrated. And this worked very well for model organisms, but can also be applied to mouse or human data. So in terms of the functional annotation, I had not really a lot of time to show you all the different details on how to perform functional annotation. I hope I could kind of show how you take different source databases like Interpro, KEGG, and Blast-based annotations and could merge them into one big project and check there and use like the power of combining different sources of informations. And this is kind of the gold standard for functional annotation as well, by following the Blast2GO methodology, you know. And there, like when working with the side panels, you can really easily combine different analysis steps, and each individual step offers a lot of different rich visualizations to review your intermediate results, but also the overall functional interpretation and visualization of functions is kind of possible here. And that's kind of all for today. My 1 hour is kind of up. I'll just stop sharing. So